My name's Blake Wills and I'm the CEO at Microba. We are a precision microbiome science company and we believe we have leading technology to analyse the human gut microbiome that can in turn be used for therapeutic candidate discovery. As you know, the microbiome has been linked to a broad range of disease conditions over the last decade. At Microba, our core thesis is that precision measurement of the human gut microbiome is required to solve unmet medical problems. And we at Microba have the globally leading technology to analyse the human gut microbiome. What do I mean by that? When we take our shotgun metagenomic analysis platform and apply it in a real patient setting, we see more of the human gut microbiome, we avoid false positives, and we also avoid false negatives. Here in a single US patient sample, we're reporting 183 species of bacteria in this disease patient, compared to the leading competitor who only shows 81 species. If you would like to learn more about that, I strongly encourage you to read the presentation, the publication I have there on the right-hand side, recently released in Frontiers in Microbiology, called Evaluation of the Microba Community Profiler. At Microba, we have an end-to-end platform to measure the human gut microbiome. That includes sample collection in a very stable and robust device. We then have optimized laboratory protocols, which have been robustly tested to ensure that we're collecting the right data all the way through to DNA sequencing. Once the sample is sequenced, we use our world-leading bioinformatic pipeline and tools to analyze millions of fragments of DNA to reconstruct an accurate picture of the human gut microbiome. And we're able to deploy this anywhere in the world with our cloud-based infrastructure. And we also leverage big data, AI, and machine learning in identifying disease-relevant signatures or biomarkers. This technology was developed by Professor Phil Hugenholtz and Professor Jean Tyson. They're experts in the field of the microbiome. They published some of the seminal work in the field during their time in the US at Berkeley, MIT, and the Joint Genome Institute. And many of their open source published tools are in fact still in use today broadly in the field. They moved back to Australia in 2010 where we set up Microba. And since then, we've developed the technology that Microba is leveraging today. The team also includes Professor Ian Fraser, inventor of Gardasil, the cervical cancer vaccine, which is now a Merck drug. Associate Professor Lutz Kraus, a leading expert in the field of AI and machine learning, previously with Nestle in Switzerland, studying the human gut microbiome, as well as a cancer genomics career. And then a broad range of commercialization and pharmaceutical development experts, Dr. Terry Ann Koch, Dr. Caroline Popper, Dr. Purako Kuv, just to name a few. As a team, we have 48 staff. We operate globally. Our IP platform is growing significantly, and we're well supported by an institutional investor base. All in all, we are global with US partnerships, European partnerships, Asian partnerships, and we're certainly excited to be presenting at this conference here today. Our commercial model at Microba is twofold. We have a precision microbiome platform, which we use for microbiome analysis. This is leading analysis for consumers, clinicians, and researchers of the human gut microbiome. And then we use a proprietary large data set that we've constructed over the past 5 years to look at biomarkers which could potentially be turned into new therapeutic candidates. And that's what I'd like to talk to you about today. Our scientific approach and thesis is that we are in a third generation of microbiome discovery. The first generation was fecal microbiome transplants, which really occurred over the last decade and beyond. Then we moved into a random library screening approach. But today we're entering the era of big data and AI-driven drug discovery. And that's what Microba is bringing to the human gut microbiome. Importantly, most of the human gut microbiome has never been brought into culture. That means we haven't been able to study it in our traditional ways. But at Microba, we're able to interrogate the whole microbiome through our DNA sequencing technology and platform approach. But beyond that, we actually have demonstrated the ability to pull out specific microbes into culture using genome-directed isolation techniques. This lets us access more of the microbiome and take a targeted approach to identification of therapeutic candidates. As you can see here, we start with our large proprietary human data set. We then use big data, AI, and machine learning to identify the leads and remove confounding factors. We isolate these bacteria that we're interested in using genome-directed isolation. And then we take broad preclinical screening approaches to identify the microbes with the activity that we're most interested in. Our flagship program at Microba is an inflammatory bowel disease. Here you can see one of our assets performing very nicely in a DSS-induced colitis model. We see great readings on the histology, including the histopathological score, epithelial injury, and inflammation score, which is comparable to prednisone, a potent corticosteroid. These microbes that we're using are derived from healthy human donors, and they're not genetically modified. So we think the safety profile, and certainly the data is supporting the safety profile of these microbes, is going to be very strong. In addition to that, we have a bioactive program where we're identifying the bioactive molecules that are being produced by these bacteria. And you can see in the bottom right-hand panel here, some of these microbes, some of these bioactives are having potent STAT3 activity, as well as working on certain GPCRs that we're very interested in. As a company, we have two primary programs. Firstly, a mild to moderate ulcerative colitis asset, which we'll be pushing into phase 1 trials next year. And then separately, we're launching what we believe is the largest microbiome-focused melanoma study, where we'll be looking at developing a combination therapy for melanoma patients with immune checkpoint inhibitors being the combination therapy. This is being launched out of the US, and we're very excited to be in possession of this data set so that we can take a data-driven approach to identifying new drug candidates. My name is Blake Wills. If you have any questions about this presentation, please do get in contact with me. My email is on the screen. We'd love to talk to you about partnering in the Asian region. We're very excited about existing partnerships that we have in South Korea, and we'd certainly like to work with people to unlock the opportunity that is in the human gut microbiome. Thank you.